Thanks so much, Ram, for the wonderful introduction and also for the invitation. It is my pleasure to be here, and you guys have very good, wonderful facility here, and I was very impressed with the research as well as the medical, the connection. Today, as Ram said, I'm going to talk at the engineering the interface. So we work at the interface of biomaterials. So here the biomaterials we designed to mimic the extracellular matrix of your tissue. So we can control the properties and we can understand why. So if you look at the native tissue, the same extracellular matrix can contribute to tissue repair, tissue homeostasis and disease. So our idea is to whether can we understand why subtle changes in the extracellular environment trigger the disease progression or the repair and how they involve the stem cells. And then I'm going to talk about how the same principle can be used to create smart materials, and that is the one with the healing systems. Before going into that, I'm going to summarize some of the activities that is going on in our lab. We work at the interface of bio-inspired, smart materials, and stem cell engineering. In the case of stem cells, we mainly work at a role of microenvironment or the niche on self-renewal and differentiation of stem cells or change in the cellular behavior. And we also started now looking at in the case of differentiation, we look at musculoskeletal systems. There we mainly focus on human pluripotent stem cells, that is embryonic stem cells as well as induced pluripotent stem cells. We work at MSCs, mainly bone marrow derived MSCs. We work at cartilage, bone and skeletal muscle, and now we started a little bit with the skin. We look at vascularized tissue, and especially when we wanted to implant them and use them as a replacement for the dysfunctional or compromised tissue, then we need to worry about vascularization. So we look at vascular tissue. Recently, we have started looking at cell motility from the perspective of cancer metastasis. So so we all know the cellular aspects of the metastatic cancer, but there is an environment which is involving extracellular matrix, and we are trying to understand how the extracellular matrix properties promotes invasion and what are the physical properties involved in this process, and we work at tissue fibrosis. In the case of biomimetic or bio-inspired materials, we work at the biomaterials with the defined bulk and interfacial properties, so we mimic the soft tissues, and as Ram mentioned, we work in the hydrogel field. The reason hydrogels is very similar to soft tissues. It is a network containing a lot of water. So if you take any of your tissue, and from the structural perspective, it is nothing but a network containing a lot of water. That is the jello that you see in the grocery shop. We work at the multifunctional synthetic matrices where we modulate the tissue properties. We work with smart materials that can self-organize, self-heal, so on and so forth. And we work at the multifunctional small molecule. In the case of multifunctional small molecule, the idea is very simple. In the case of a stem cell transplantation or cell transplantation, you take the cells, culture them in vitro, and then put them back. So the idea of the functional small molecule is that, can I take a pill which has multiple properties? When it gets into the targeted site, can this pill manipulate the environment? So for example, if you look at systemic diseases like Duchenne muscular dystrophy. If you look at the Duchenne muscular dystrophy, it is a cellular component is missing, that is a dystrophin. But if you take the environment and look at the extracellular matrix, you have fibrosis, you have inflammation. So when you transplant the cells, unless and you manipulate this environment, the cells are going to die. So our idea is that we have created a small molecule which can target multiple pathways, like NFKP pathway for inflammation, TGF-beta pathway for fibrosis, and also has an epigenetic component that can promote stem cell differentiation and also work at the interface of cell surface molecule and thereby avoid the friction. So you take a pill, this can do all those things, so you get better with the time. So that is the idea. Now today I'm going to talk about why we need to care about the interaction of cells with their extracellular matrix and what is the effect of the extracellular matrix. So if you look at the interaction of cells with their physical environment, which plays a role in disease and repair. So you take a disease, there is an interaction. You take a repair, there is an interaction. You take tissue homeostasis, there is an interaction. And if you look at these interactions, one component is cell matrix interactions, how the cell is interacting with the matrix through the interface. The next one is cell-cell interactions, how is the cell interacting with its own neighboring cells, and what the neighboring cells do to it. Then you have a surrounding water component. There you have soluble morphogens because your tissue is highly hydrated, so you have this aqueous environment. So the idea is that if I can control these interactions which happens at the interface, then I might be able to control the adhesion of the cells, proliferation of the cells, and differentiation of the cells, and also use it as a tool to understand the disease progression and also contribute to the regenerative medicine approaches or strategies. Now, as we all know, there are studies showing the role of chemical functional group, matrix stiffness, matrix rigidity. All these plays a role in stem cell shape manipulation and their differentiation. So the idea is, if you look at most of these interactions happens at the interface, but most of the time we miss to understand that interface, like how the interface playing a role. So if you look at the Akil Thompson's book on growth and form almost a century ago, he talks about this interface and how one can manipulate this interface to promote adhesion or cohesion, and how you can use that to control the morphogenesis. So we took a step back and asked the same question. Okay, now if you manipulate the cell matrix interface, you have the same effect, and can we use this cell matrix interface to control the cell shape, and by controlling can we manipulate the morphogenesis? That means what the cells become or how they build the hierarchical tissue. So we went back and talked about how would we engineer the interface. So when you interface engineering, one way you could do is that you manipulate the way the material is going to interact with the water. So you have a hydrated system. So you at the interface, the material is interacting with the water. So you wanted to figure out how can you modulate this interaction. And one way you could modulate that interaction is changing their ability to interact with the water, increase or decrease. So if you take a lotus leaf and drop a water molecule, you see they form a round shape, so that the water doesn't spread. But if you take another leaf and then spot a drop of water, it is going to spread. So hydrophobic when the water doesn't spread, and hydrophilic when the water spread. Now, if you want to understand how they interact at the interface, then you wanted to make sure that there is no change in functional group and there is no change in matrix stiffness. So what we did is that we went back and we devised a copolymerization strategy from the polymer principles. So we have an acrylamide because you can use any monomer you need. We used acrylamide just because everyone uses acrylamide, so it's an easy system to work. Now we created this material. The beauty of this material is that I can increase the length of this material by changing the CH2 group. I can go from 1 to 10. So what I'm doing is that I have a network and I'm introducing this hand, and by changing the N, I'm increasing the length of the hand. Now when I do that, we can see that this is hydrophilic and this is hydrophobic. This is the water droplet on the surface of these materials, and you can see the water contact angle is changing. So you know that now the surface property is changing. Now in all these cases you have the COH group, so we are keeping the functional group the same, and the matrix rigidity is pretty much the same, so we are not making any difference in the bulk rigidity. So we have a tunable matrix with varying interfacial hydrophobicity or hydrophilicity. We look at the topography, they are also surface roughness is minimum, so it is a very smooth surface, and so the surface roughness has nothing to do with the cells. Now when you take these materials and you add the cells, and I'm going to call them C1, C2, C3, C4 because it is just the CH2 number of CH2 groups. Now when I introduce, this is adult bone marrow derived mesenchymal stem cells. When I introduce the stem cells on top of these materials, you can see when I increase the hydrophobicity, more and more cells adhere. Now if I increase the hydrophobicity further, less cells adhere. So you can see they have a non-monotonic dependence or a bell-shaped curve. Now if I look at the cell shape, I do see somewhat similar. The spreading is also very much dependent upon what is the interfacial hydrophobicity. Now if the cell adhere and spread more, that means they are adhere more to the material. So you need more force to detach the cells. So we went back and devised a shear chamber, and we exposed the cells to shear force and looked at the force required to detach the cells, and you can see you need more force for the cells from the C5 to come out. So that was anticipated. Now I'm just going to summarize this slide saying that this is nothing to do with the monomer, because we change the monomer, and if you look at, still we see a non-monotonic dependence. So that is this just proves that it is the hydrophobicity at the interface is playing a role. Now this is the cells on C3, and if I look at the cells on C3, you can see the cells move like a slippery surface. They are moving on a slippery surface. They don't have a strong adhesion with the surface, so they are more like a rubber band. They just stretch back, so they don't like. And whenever, if you look at here, whenever the cells comes in contact, they like to interact with them rather than spreading onto the material. So they affect their mobility, they affect their shape, they affect their ability to interact with other cells. Now if I go to the next material and I look at the C5, this is where you see more cells adhering, and you can see the cells are very adhered to the material. The cells don't like to move that much because they are strongly adhered to the material, and they have a directionality. They don't slip or they don't jump like that. And whenever if you follow these cells and plot them, you can see that in the case of C1, C3, what you see is that they are doing a random walk model. They are randomly moving on the surface. There is no directionality. And this is called a random walk model. The reason this is called a random walk model, that is how a drunkard man walks after getting drunk. So if you follow the footsteps of a drunkard man, this is actually what you see. So there is minimum adhesion, so the cells are just randomly walking. Now if you take the cells on C5, you can see they have a directionality. One cell follow the other cell, they go in one direction. And if you look at the path length or the distance traveled, and you can see that the cells on C1 travel more because they are randomly moving, there is no interaction. And in the case of C5, they have strong adhesion to the material, so they have to lag.
Is the cell density matters? Or?
It doesn't, because this is in the case what happens is that they can proliferate more on C5. So you see that, but then we decoupled that, so it is more the adhesion. So we have a movie from day 1 to day 4, and what I'm showing is day 4, because day 1 you need to have a microscope, like very, you have to zoom it up a lot to see the cell movement.
Is there a reason why they are directed in motion on C5?
So one of the feeling that I have is that there is something to do with integrin addition, and we see some pattern. But it is very difficult to decouple because focal adhesions, like whenever they attach, there is a focal adhesion in the other one too. So we are now doing some experiments to see with the vinculin to see how they are interacting with the material. So we can see the tensor, like where the force is focused.
Did you do any experiments extending this question? ICAM, that's intracellular adhesion molecules, which could be present in these adhesions.
So in the mainly it is the cell matrix addition rather than the cadherin molecule. So we looked at the cadherin molecule, but the problem with here is that these cells also differentiate. So in the differentiation you are going to see some pattern, right? So that is why I was saying that vinculin is always common. So now my Martin Schwartz group has developed that vinculin tensor, so we could just put them and then look at them how they move. So now a graduate student is working on it, developing the system. Now if I take the cells on C3, this is at day 4, and you can see they are very motile cells, and if you culture them for a long time, this is after 21 days, and you can see these cells cluster and they form a rosette-like structure. They like to interact with the other cells. And if I take the C5, this is after day 4, and you can see there are more cells because they proliferate, and there are stress fibers. And if I look at them after day 21, you can see they align. So they take a different stress level and a different shape. Now if I take these cells from the growth medium and I try to differentiate them, what happens is that the cells just die. They don't like to. So these cells lack the ability to reorganize their cytoskeletal structure. At least that's what our speculation. So that's why they just died, because they detach from during that process, they cannot adhere back and they die. In this case, depending upon the cell shape, we can differentiate them into bone cells or the muscle cells. So if I take this and leave them for a long time, they become osteoblasts, and if I withdraw the serum from this, they become myoblasts or myocytes. I'm just taking the day 4 cells. This is after day 4. Put them in osteogenic medium. They undergo osteogenic differentiation. ALP is an early osteogenic marker. Alicerin redus is the extracellular matrix, collagen type 1, and osteocalcin is the late marker. And you can see they also express the gene expression level. Now, if I don't add them into osteogenic medium, and if we leave them in culture in growth medium, it takes 21 days for them to differentiate, but they ultimately differentiate into osteogenic phenotype. By introducing the osteogenic medium, we can speed the process.
You don't see any kind of cell death or anything?
Whenever you have a lot of cells, there is always going to be some cell death. That has nothing to do with the material. Even in plastic, you will see. The reason is that the crowding effect, so the cells has to that. But the cell death is very, very minimum. Yeah. Now, if I take the aligned cell culture and withdraw the serum, in the case of myogenic, if you culture them in a BFGF, so because in the higher FBS, they always proliferate and they don't undergo myogenesis, so you need to withdraw the serum. And if you do that, they undergo myogenic differentiation, positive for desmin, myosin heavy chain, and MF20, and again you see the same from the gene profile. So we can just take the cell shape and switch the culture condition, you do get a myogenic differentiation or osteogenic differentiation. Then we asked the question, why do we see this? Now, when you increase the chain length or the dangling side chain from C1 to C7, what we are doing is that we are increasing the hand length, right? So now when at C1 or C3, my hand is not long enough to come to the interface. So they don't bind to the protein or the cell secretory factors. So I have an issue with the C1 and C3 because that is not long enough. Now when I go to C5, my hand is so long enough that it can come to interface. So they bind to the protein. Now if this is true, then I should see the same effect with the C7 because I'm increasing the chain length, right? But what happens in the case of C7, my hand is so long, it is so flimsy now, and it is also so hydrophobic, they flip. So I hide the COH group. The functional group is now hidden, so they cannot bind or interact with the interface. In fact, we prove that using molecular simulations. And this is a solubility curve. You can see C5, they are at the interface, C7, they are collapsed. They cannot interact with the water, so they just collapse back. Now, we also proved that it is not only about the protein binding, it is also the conformation. If it is only protein binding, by increasing more C5, I should be able to get more differentiation, but that doesn't happen. So we know that it is more than protein adsorption. What we need is the conformation, like what is portraying onto the surface to the cells and how they can interact with the cells. So after proving this, we want to ask a question, can we use the same strategy to create materials or tissue cultures for embryonic stem cells? Now the question is, why do we want to do that? One issue is that if current cultures, if you look, they all are cultured on the MEF, that is the mouse embryonic fibroblast. So whenever you talk about these pluripotent stem cells, that is embryonic stem cells or induced pluripotent stem cells, they are a spoiled brat. They don't like to live on or sleep on or grow on a plastic. They want another layer of cells or a very good mattress to live. So now you give the mattress, that is mouse embryonic fibroblast. So you can see this is the mouse embryonic fibroblast, and these are the colonies. So this is the embryonic stem cell colony. So they culture on top of the mouse embryonic fibroblast. You can also culture these cells on top of metrigel that you get from the tumor of the mouse cell. So you grow a tumor in mouse, take the tumor, extract the extracellular matrix protein, and you culture the embryonic stem cells, they do grow. Now, with the CIRM funding and California Initiative on Stem Cell Research, if we want to take these cell lines into clinical trials, there is a big problem. Whenever we culture the cells on these mouse-derived proteins or mouse-derived cells, and you analyze these human embryonic stem cells, what happens is that the mouse embryonic stem cells secrete certain proteins or a sugar molecule that go and contaminate your embryonic stem cells. In fact, this is a sialic acid molecule, and in fact, Ajit Varghese group has identified this molecule, and they noticed that both in the MEF and the cells cultured on metrigel, the cells are always contaminated with this animal-derived protein. So you cannot translate the cells cultured on this material into a clinical trial. That is one issue. The second problem is that you need to, when you talk about clinical applications, we are talking about, or even drug screening applications, we are talking about scaling the material. So if you wanted to, you want millions and billions of cells. Now, if you consider MEF or Metrigel, it is very hard to get that many cells because it becomes very expensive. Metrigel is very expensive. MEF, one vial is, irradiated one is, what, $16 or $23. Then you can only culture 3 vials, 3 6-well plates. So you need more and more materials, and the cost adds up. And also batch to batch variation. So if you take, you always have to make sure that the load is the same, the host animal is the same, so there is a lot of variability. So it is very difficult to scale them. So we asked a question that, okay, can we take them into the lab and see whether we could create a plastic where we can grow these cells. Now, if you look at the human embryonic stem cells, there are one thing it is required is BFGF. You need to kept on adding a lot of BFGF molecule, otherwise they change their phenotype. Now, when we ask the question that when you give to the BFGF, one of the molecules that regulate the BFGF signaling is heparin. So, and Jamie Thomason's lab showed that MEF, the embryonic stem cells, secrete heparin, and that regulate this BFGF signaling. So we went back to the lab and asked a question, can we create a protein or a polymer that is very similar to heparin in docking the BFGF? So we asked a simple question. So we took a PSS molecule, that is a polystyrene sulfonate, and notice that the BFGF docking of this molecule is very similar to heparin. So we came back and we made this material. You don't need to know, then we just did some polymer chemistry. And we made this material. So this is, we call it as a synthetic mimetic of heparin. Now we also use the same copolymerization principle. So we can vary the mole ratio, we can vary the rigidity, hydrophobicity, chemistry, functional group. So we can understand all these parameters, how they affect self-renewal of embryonic stem cells. And this is a picture after 20 passages, that means 9 months and 3 weeks, these cells on this material. And you can see they are positive for NANOC, they are positive for OCT4. These are the main pluripotent markers, and they support in stem pro medium. And we need to do in stem pro medium because this is a defined medium. So there is no animal derived products, and you can see the cells are beautifully on these cells and they grow. This is just showing that they are all the properties are comparable to Metrigel. This is quantitative PCR and facts. And we look at one other characteristic of embryonic stem cells is that they can differentiate into all the 3 germ layers, ectoderm, mesoderm, and endoderm. So we went back and looked into their differentiation, and they differentiated into all the 3 germ layers. They are karyotypically stable even after 9 months of culture. And we looked into other cell lines like HUCITs, iPS cells, and we can mimic the same attributes into these cells. And these are, you don't need to worry about this, is we are characterizing the rigidity. We are looking at how the hydrophobicity, and we can only get in when they have a hydrophobicity of 23 or hydrophilicity of 23, and this rigidity, the cells exhibit the colony formation and pluripotent phenotype. If we disturb that, the cells don't differentiate into, or they don't stay as an embryonic stem cell.
So what you have growing?
Is a HUS9 embryonic stem cell. Oh no, so in this one was just a differentiation showing that they can differentiate. Now this was just a differentiated product. In this case it is just, yeah, it's a pluripotent stem cell growing. So then we proved that they can grow for 20 months, 20 passage, that is 9 months. Then we took this material to understand how the interface play a role, how the chemistry play a role, what is the stiffness and things like that. So here it is pluripotent. Yeah, but we killed it. It's very expensive. Now we are trying to submit the paper and move on because it gets very, very expensive because it's a stem pro medium. So the one bowl of medium is $500, so it gets very, very expensive.
Maybe you should synthesize that.
We also now wanted to figure out the carboxylate group versus sulfonate group. That means you have a COH group and you have an SO3H group, and this is one and this is another one, and we know, and we matched every properties, and we know that it works only in the SO3H group. So it works only when a sulfonate group. And if you look at the beauty of heparin is, it is a sulfonate molecule. So it was intuitive, but we want to prove it, so we went back and re-engineer everything, and then or redesign the material, and then we proved that it's endued through SO3H groups. This is just showing that what are the endogenous markers expressed on the cells, and the take-home message from this slide is that the material provides some adhesive sites, so the cells can adhere. Now and this provides an environment such that the material starts secreting its own proteins. That is, if you look at fibronectin, laminin and vitronectin, they are relevant for embryonic stem cell self-renewal, and they organize them in such a way that they create a niche and engage the corresponding integrins and promote self-renewal. So that is what actually our theory is saying, that okay, this is what it happens. Now, okay, we can self-renew the cells. We can do that. So in fact, we are trying to license this out now with in vitrogen. Now, the question which we asked is that can we use the same principle or design principle to create materials that can now contribute to repair? So if you look at the bone in your hands or your teeth, this follows a biomineralization principle. So it binds to a mineral calcium, and then it allows them to form these beautiful structures. So our first question, that can we take our hydrogel, which looks like a very transparent, it's like the condyle lens that you have. Now if you allow them to undergo the biomineralization, then you can get a very opaque tissue. And if you look at their structures, this is more like an apatite-like structure. That means what you have in your bone tissue. And then we wanted to ask a question that we can use this material to promote differentiation of stem cells. This is with the bone marrow derived human mesenchymal stem cells, and you can see that they upregulate runners 2, bone protein and osteocalcin, the markers of osteogenic differentiation. We look at biochemical assays, and you can see they upregulate ALP activity, and also you can see that in the mineralized material or the biomineralized material, they express osteocalcin, which you do not see. That is a late osteogenic marker, and in all these cases, we use growth medium. So we do not add any growth factors, we do not add any soluble morphogens. This is a cell which is in 10% FBS in DMM, high glucose DMM, that's it. We then took these materials, put them into a rat, and then looking at how the ectopic bone formation, and you can see this is at day 1 or day 2, and you can see more opaque tissues. They are ectopic bone formation. This is micro CT. We are looking at how the bone form, and what we did here is 3 things. So this is a mice model. So we took human bone marrow derived mesenchymal stem cells, transplanted, and then injected them in, so put them into the material and then transplanted them into the subcutaneous into the mice. Now this allows us to have a real-time imaging to know the viability of the cells. So this tells us that the transplanted cells were viable and they proliferated and they were living there happily. So this is where you can see, when we open up, you can see this is vascularized. Now the second thing we did is that we took a material, but there is no cell. And if you look at it, they are also highly vascularized. Now if you do the same material, if it is not biomineralized, you can see there is no tissue formation, it's still opaque. Now we took this tissue and did the histopathology, and this is the middle section, and you can see the material is highly vascularized, so it is not on the surface, and they also form bone tissue. So we don't even need the cell anymore. We just need to take the material, put it into the defect. The material recruit the cells from the patient and promote their osteogenic differentiation and also vascularize. In the case of a bone tissue, it is highly vascularized. So if the material doesn't vascularize, then you are having an apoptotic tissue. So most of the times what people do is that they add growth factors to vascularize. We don't need it. We can just need to put the material, and this is, you can synthesize what, $5, 10 cents, I don't know. So you can make this material easily, and they are biocompatible, and they don't encapsulate into the capsule. There is no macrophage interaction. There is a slight inflammation initially, but that contributes to the vascularization.
How is it acellular? So you started with
the material, just the material, put it without the cells.
Get the mineralization, did the cells drive?
No, in this case we cheated that, so we took the cells out. So the mineralization is done by chemical process. So we mimic how the mineralization happens in the in vivo, and we developed a stimulating body fluid. So what we did is that we have these hands. So this COH group can bind to calcium. And the calcium is presented in such a way that the phosphate groups comes and forms this beautiful apatite structure. So we can replace the cells by this molecule. And then when we put them into the in vivo, this material undergoes a dynamic dissolution. It acts like the bone, which is act as a reservoir for calcium and phosphate, and that signaling promotes recruitment of the cells as well as vascularization.
How does that compare with taking, let's say, ex vivo bone?
So right now we are having a posterolateral fusion model going on. There we are using autograft as a control to see how good this material is. So I don't have a data to tell that, but we are doing that now. Because we went with, in the cranial defect, it looks very similar to the this one, but you know cranial defect can heal automatically, so I don't know whether that is a representative. So we were now doing posterolateral fusion as well as a femoral defect.
So how is that, a lot of people have tried for many years to come up with replacement materials for bone and for other soft tissue,
Yeah.
hard and soft tissue. So how does that differ from how other people?
So what people, most of them are native polymers, like collagen. Most of them are collagen, right? This is totally synthetic. The idea, so the synthetic makes us to have a knob how to control the degradation. So we can control the degradation of this material anywhere from 3 weeks to 7 months.
So ultimately, bone, depending on where it is going to have long fibrous collagen bundles for structural stability.
Yeah. So we do see collagen infiltration and deforming. So we did not do any the structural organization. We only stained them because this is always subcutaneous data. Now we are trying to look at with micro-CT, TM, all those to look at hierarchical organization and see. So we do see the fiber formation in fact in SEM. When we do the SEM, we do see, but we didn't look at how much the bundles and things like that. And also if you look,
Because you've done a lot of work with orientation.
yeah.
And so do you think that those fiber bundles will orientate themselves in a way, or do you have some ideas on how to do that?
So in the case of this one, since we are putting the material inside, it is very hard. So in the case of an orientation, you need a stress or the force in that direction, right? So in this case, it is an anisotropic material. We can always control the force by controlling the rigidity of the material, by changing the spatial degradation. But we didn't go to anywhere there. No, we haven't yet. Right now, we were very much into finding out the mechanism, because it was very surprising to us that they endogenously recruited all these cells. I wanted to do more like a biocompatible to the material. So my idea was more like, okay, let us put it into the animal and see whether the acellular material is biocompatible. Because in this case, it is HMSC, so cell might be playing some role.
But these are aphymen.
So this was a nude mice, but this one is from a wild type mice.
So is there a substantial inflammatory response?
We did not see. That is where we want to figure it out why it's not happening. So that's why we went with the mechanistic study now. So we do see initially, because we bleed the cell, because when we do the subcutaneous, there is a bleeding. So we do see 24 hours of inflammation. So it's not like that zero. But afterwards, there is no inflammation. And we don't see they're getting encaps in the capsule. There is no capsule formation. You do see a thin layer initially if you do 3 days, but this was after a 4 to 8 weeks, you don't see anymore. Yeah, so we went into the mechanistic study to figure it out, what is going on and things like that. And then we started this posterolateral fusion model, so that's a big study going on. We have some idea where we think that phosphate, transmembrane phosphate transporter plays a role in signaling the differentiation. We have some data showing the role of ERK, PP38, and also now we are finding out that there must be some influence from mitochondria because ATP, but then I don't have any concrete data either way, so we are just doing it now. We also create so if you look at all these materials, they are dynamic at a certain chemical level, but if I wanted to, if you look at your tissue like a skeletal muscle, which contracts, expand, so there is also dynamic in the mechanical level. So we asked a question that, can we create bioactuators? Just not having a material which is only dynamic in the chemical level, can we also make it a mechanical level smart or actuating type? So if you look at, this is a sea cucumber. So if you look at the sea cucumber, when you take them, touch them, they start expelling water. Now if you remove the hand, they take it back. So the idea was that, can we create materials that can oscillate? So in this case, we have created a material which can oscillate between this is initial material which we developed. Now we can oscillate between 35 and 38 or 34 and 37. So they swell and collapse, and so we oscillate in between these two. So we have, this is just the chemistry, this is the entire device. So we need temperature fluctuations. So in this case, you heat them at 35, cool them to 32.5, so they oscillate. So this is between the 32.5 and 35.5, so you're oscillating by changing the temperature. So that is why you need this device, cold water, hot water, and you just control them with the computer. This is, in fact, there is a movie showing, so it shows that they oscillate within, we were tracking the particles within the hydrogel, and this is a trajectory. If you don't have the similar chemistry, you don't have an oscillation. Here you have an oscillation.
So is the oscillation actually steric conformational change?
No,
The
in the volume. Water going out, water coming in, so the change. So we can get a volume strain. So there is an oscillation, and if you look at osteogenic differentiation, the oscillating materials promote osteogenic differentiation compared to control. I did something, I don't know. Yeah, so the right side, they promote the oscillating material that promote the osteogenic differentiation, and control won't. So now we are perfecting the device, because then we can control them multiple time and look at them, how much strain you need, what are the strains, and how the cells respond to this strain or sense the strain, so on and so forth. Then we also look at tissues like a neuron, tissues like skeletal muscle. There is also a voltage potential, electrochemical, electrical potential. So we have created another hydrogel which is straight at some point, and if you expose them to electric field, they bend, and we can change the directionality of this bending, and we can make them left, we can make them right, circle, and so this is a device.
That's by switching the
electric field. So you on and off switching. So you can control them, they go back. Now you have because they bend, you have a mechanical cue, and now there is an electric field, so you have an electric cue, and also you can play with the chemistry, so you have a chemical cues. So we call it a 3-in-1 or trinity in one. So you have chemical cues, you have mechanical cues, and you have electrical cues, and all of them can be dynamic because we can switch on and off. And the device is very small. It's in fact, the first author of this paper is an undergraduate student, so that's why you can see the device is very much custom designed to the lab. No optical control. Yeah, that is another one, right? Now we culture the stem cells, and it is cell compatible, so there is no cell death. And also in this case, we looked at their differentiation to cartilage, and we know that this promote cartilage differentiation, or chondrogenic differentiation, and yeah, so they support for 21 days. We only did it for 21 days, and promote chondrogenic differentiation, because that's the one pathway we were looking into. And now the student is again trying to figure it out, how that happens, how can we decouple each of these cues and look at which one is more important than the other one, okay, without the optical cues. Now this is the work which Ram was talking about. Now if you look at all this, I've told that we work with the hydrogel because they are soft tissues. So we call it as a mimicry of soft tissues. So if you take your tissues, skeletal muscle, that is a network of proteins with water. So in this case, if you look at it, they are also a highly water-filled network. So the water doesn't come off, and they absorb them and stay there. This is very different than the sponge. So this is not a sponge. Now, if you look at this is a chemically cross-linked network, so the molecules are all constrained. So they cannot move around. So you cannot heal them. Now, if you take a native tissue, you cut them, they can heal. So one of the things which take this material, the second layer citizen, is that they cannot heal like soft tissues. So we asked a question, can we make them? We can make them to change to the environment They can respond where they touch, they move. We can do all those things, but we cannot heal them. So we asked the question that how can we heal this material? So we said, okay, now we have so we created a material. I'm going to explain this later. They can heal, and you can see we can kept on healing them, and they just kept on growing. You can take, this is a 3 pieces which we healed, and you can see that we can stretch them, and they don't break in the interface. So the healing phase is one here, and you can see they don't break there. Now I'm also going to show you the healing, and then I explain how it happens. So if you look at this, we colored them just for the visual appealing. So they are the same material, one we colored with red and one with yellow, so we can see it. So this is a pH 3 buffer, and this is a pH 7.4, and you can see that they heal instantaneously. It takes less than 2 seconds to heal, and he kept on stretching them, and this does not separate. And if you go to my website, there is a YouTube where he describes this process. Now, despite repeated stretching, the hydrogels can still stick to each other. But now if we take the hydrogel, put them at higher pH, like pH 10 and 9 that we did, and they come back, they separate at the interface. Now, how would we do this? Now, whenever I talked about the hydrogel, this is how the hydrogel looks like. And you can see this is the backbone. And now what we did is that we attached these arms onto the hydrogel, and this is the arm. So you can see that there is an arm, and we change the N, and thereby we increase the length of the arm. So that's what we are doing. And this is the cross-linker, so we cannot separate the material. Now we can increase this arm by changing the material. So you can see that when I increase the CH2 group, so I can go from 1, 3, 5, 7, and 10. So I'm increasing the length chain. Now we can only heal the material when we have this length, that is the 5 CH2 groups. Now what is happening is that at a pH above 10, these materials are CO minus. That means you have one negative charge here and another negative charge here, and when they come together, they don't like it because they are the same charge, they repel. Now so there is an electrostatic repulsion. Now when we change the pH to 3, the groups get protonated. Now they don't have the negative charge. Instead, the negative charge pick up a hydrogen and make them COH group. Now when we expose them together, they interdigitate. They interdigitate using hydrogen bonding. So now you have a interdigitated molecule where the C double bond OH interacting with this amine group, or the OH group interacting with the amine group. We proved this using Raman spectrum, FTR spectrum, so on and so forth. And you can see that they are interdigitated. And in order for this to have the interdigitation, you need the right length. So I have these fingers, they have to come like this, and they grab like this. That's why it's instantaneous process. Now if they don't have the right length, then they cannot grab properly. So you don't want it too small, then the interdigitation is not perfect, and you don't want them to be this much, then they cannot interdigitate. So they have to be the optimal length. So this is the collision day principle. You have to be at the same time at the same place. Now we proved this using computer simulation. This is what previously I showed you, that when we increase the length, we are flipping the chain will flip, so it is not accessible. So we did some simulation real time to show that indeed that happens. In the case of an ACHAC, you can see this blue, that is the amide group. They are accessible, so they can bend. And you can see it is interdigitation because you need the accessibility of the amide group to form that interdigitated structure. Now we want to find some application for these materials. One application we thought is that if you have a scratch on your car, you have to go to the body shop, you have to repaint it, and it costs a lot. So what if that we create a plastic or a paint which can be self-healed? So we made a cut. So you can see that there is a cut here. So we painted them and made the cut, and then you sprayed the material into this cut and sprayed the pH 3 buffer, and you can see that it healed, it joined it. Then since it happens at pH 3, the graduate student who was Amaya, he had an idea of carrying acid around the lab. So he took a centrifuge tube, cut the bottom of the centrifuge tube, plug the material into it, and pour the acid, and you can see the acid doesn't come off. And so you can use it as a sealant when you wanted to especially the transport, chemical industry is a big problem. If there is any leakage, always acid spill. So you have to clean it up and things like that. So now we have a coating which seal the material immediately. So this is more for a leak, not for a big hole type. Then we said, okay, we need to find now our biomedical applications. So we took the rabbit stomach, because you know the pH in the stomach is acidic. So that is a gastric issue. So we took the rabbit, and this is an S plant. And then we adhered the material into it. And this material was also loaded with the cyclode. Okay, I forgot the drug. So the tetracycline, and we loaded them with the tetracycline, and we looked at the release profile. This can adhere to this one. You can change the pH. You can take the thing out. And this is only done with S plant, and this is not an in vivo study. So the idea is that can you inject the material into the perforated stomach and stop the perforation so the gastric juice won't be leaked out, so on and so forth. But we haven't explored any of them. So in conclusion, I hope I convinced you guys that we can engineer the interface, and interface is very important. And by engineering the interface, we can control the cell matrix addition or the cell shape. We can also control the interface to increase the cell-cell interaction, because like embryonic stem cells, they need to grow as a colony. So you don't want a material which pull the cells and spread them. You want them to be in the right shape so they can adhere with other cells. So cell-cell addition. So you can regulate self-renewal versus differentiation, or proliferation versus differentiation. We can create synthetic material that can be used everywhere for a self-renewal of HPSCs. So you can create millions and tons of these cells, then you can differentiate them, so you can use them for drug screening. I don't know about using into the human, but at least for the drug screening platform. We are very excited about the synthetic bone graft. Now we have a cheap alternative to allograft, or we don't need BMB. The current market, 70% of market is with Medtronic. They have a infused system. There you know they have the BMB. So the BMB secretion or the movement of the BMB in the tissue can cause other problems like ectopic bone formation. So here we have a system which does not release any growth factor, nothing. It can direct differentiation and form vascular tissue. More than any of these things, as a basic scientist perspective, this gives you a platform where you can tune all those. So you have multiple knobs, you control the knobs, you vary the knobs and see how these knobs interact, how these cues interact, or which one is important for the cellular commitment versus function. If you look at in the tissue, in the body, we all have collagen, fibronectin, polysaccharides. So the only thing which varies is the structure or the composition or their organization. So can we know how this organization changes in the cancer versus in the living, in a normal tissue? And finally, I think we can use the same approach to create materials that can be very smart, or at least mimic certain functions that we have in the native tissues. Most of this work was done by my group, and my collaborators helped me all in brainstorming and things like that, and the funding from multiple agencies, and I would like to take any questions.